Hi everyone. In this video, we are going to understand different chemical changes that alters the regular Watson-Crick base pairing in DNA. So basically, we know the structures of the purines and pyrimidines, how that purines and pyrimidines form the base pairing and everything. So coming to these purines, so we have adenine and guanine as a purines, and these purines have two rings in their structure. That is very known to us. So when we look into the adenine and guanine, so there we find some functional groups. For example, amino group is present in adenine. At the same time, we find the oxo group in the guanine at the sixth carbon. So in addition to that, in the purines, in the guanine, there is another additional functional group, which is amino group that is present on the second carbon. Okay. So some chemical changes like deamination, for example, in adenine, the deamination, the presence of water makes or replaces the amino functional group at the sixth carbon with the oxo group, so that this adenine in generally we know that it pairs with the thymine. Okay, so when this adenine forms or undergoes this kind of chemical change, so this adenine becomes into the hypoxanthine. So that means the adenine becomes into a abnormal base. So when we call the abnormal base, means that these bases are not usually or naturally present in the cells. So adenine converted into the hypoxanthine by undergoing the deamination process in the presence of water. So in this process, this reaction also produces ammonia as a byproduct. Okay. Similarly, when we look into the cytosine, in the cytosine also, where the sixth carbon atom has the amino group, though it is a single ring having structure. So this single ring has basically, sorry, not at the sixth carbon, so fourth carbon it has the amino group. The same amino group undergoes the similar type of deamination process, and the presence of water, and this amino group is replaced by the oxo group, so that the cytosine is converted into the uracil. Okay, so adenine converts into the hypoxanthine, and cytosine is converted into the uracil. So both this hypoxanthine and uracil both are abnormal bases. So these abnormal bases are easily identified or recognized by the DNA repair system, and eliminates these abnormal bases from the DNA sequences. So this kind of deamination is a very simple phenomenon which is observed in the DNA during the chemical changes. Okay. Also, we know that these purines and pyrimidines, they undergo some small chemical change, like structural change. So that is tautomerization. So during the tautomerization, so the proton from the amino group is transferred to the other atom on the same molecule. Okay, so this proton shift is called this proton shift also affects its pairing. So usually adenine pairs with the thymine, we know that. So because of this conversion of this change of the proton location on the molecule, so this adenine becomes into that means this amino group becomes into imino group. So that this amino form will be converted into imino form, and also during this process what happens, this particular adenine, so it usually pairs with the thymine. So now it pairs with the cytosine. So this adenine pairs with the cytosine. Okay, and similarly when we look into the guanine, so when we look into the guanine, what happens, in case of the this guanine, so usually this pairs with the cytosine. Guanine usually pairs with the cytosine. So instead of pairing with the cytosine, this guanine pairs with the thymine. So when it pairs with the thymine, so when this guanine, when the proton from its nitrogen atom moves to the functional group, so this keto group becomes into enol group. When the guanine keto group becomes into enol group, So what happens in this no this guanine pairs with the thymine instead of the cytosine. But see, this type of pairing is a not a abnormal. So these bases are not abnormal bases. So this amino form and enol form bases, enol form, amino form, immunoform, keto form, and enol form bases are not the abnormal bases. Unlike where the deamination of the that adenine or cytosine that produces hypoxanthine and uracil. Okay, so that irregular or abnormal bases can be easily identified, but when in a DNA sequence, because of this tautomeric shifts, okay, while producing the nucleotides on the daughter DNA, so a wrong nucleotides may be incorporated. For example, adenine in presence of in its immunoform, so rather than having as a thymine as its as a partner base on the other strand, the growing DNA strand, so it may have the cytosine, so that causes or that leads to the mutations in the DNA. Okay, so the chemical changes they cause this type of the chemical changes in bases that causes changes in the DNA sequence. So apart from this, so we find sometimes on the DNA that some nucleotides like no thymidine, which may present in a sequence. So when they are present adjacently, so these thymines, so they form covalent bonds between them. So usually when we look into the DNA structure, there we know that bases they don't have any chemical interactions or chemical bonds between them, other than small electrostatic or van der Waal interactions between them. So but when these bases which we know that they link to the sugars, and the sugars are further joined by phosphodiester bond. So that backbone of the DNA is present between the sugars, not between the bases. But so sometimes these bases they may form that covalent bonds. So when they form covalent bonds, so the topology of the DNA changes. That means the number of nucleotides or the major and minor groove structure. So that may affect by this dimerization of the nucleotides. Okay, so these are the some ways that DNA incorporates the damage in its sequence or in structure. So in addition to that, some chemical agents some chemical agents like bromouracil is one such thing. So this bromouracil, which actually in its this keto form. So when it converts into the enol form 5-bromouracil, so when it converts into the its tautomer form, so it uracil this pairs with the guanine. So usually this one must pair with the adenine actually, but instead of pairing with the this adenine, so it pairs with the guanine. So when it pairs with the guanine, uh, so this creates the change. This brings the change in the DNA sequence, or it causes the change in the DNA sequence. So therefore this type of base analogs like 5-bromouracil, so this type of base analogs, so this actually thymine analog. So thymine structure, so it's similar to the it is everything is similar to the thymine, except that that bromine is present on its fifth carbon atom. So when the fifth carbon atom is alkylated by the bromine, so this thymine analog, that 5-bromouracil, this particular one pairs with the guanine. So so that when this kind of base is present on the template DNA, so this incorporates the guanine in the daughter DNA instead of the adenine. So that results in the sequence change in the DNA of the daughter strand. So this type of agents also they cause the change in the DNA sequence. Okay, that means so usually this particular type of base analogs are not present in the cell. Naturally, they are not present. So these type of analogs are used to create the to study the mutations in the organisms. So to study the mutations, to the DNA must undergo the change in its DNA sequence. So to create such a changes in the DNA sequence, so this type of base analogs are used. So additionally, so some other chemical agents like ethidium, proflavin, and acridinarine. So these chemical agents they bind to DNA. So they bind to DNA. Where they exactly bind to the DNA. So they don't bind between the sugars or not. They don't bind to the phosphodiester bond. So they bind in between the bases of the same strand. So they bind in between the bases of the same strand. And when they present in between the bases, so when the replication machinery it reaches to the place where this type of chemical agents present between the bases. So the replication machinery may read this chemical agent as a one nucleotide present on the template DNA, and it may tries to incorporate a nucleotide on the daughter DNA against this particular chemical agent. So that leads to the frame shift of the DNA sequence on the daughter strand. So thymine dimers are one type of DNA error, DNA damage. The thymine dimers are one type of DNA damages, and this base analogs and this type of chemical agents, they are one type of DNA damages. And apart from these, as we said, some deamination process. So deamination of the cytosine or the deamination of adenine, so they also produces some damages on the DNA. So when we look into the DNA, so sometimes even we find some cuts on the DNA, so that also damage on the DNA. So sometimes cut may be present on the single strand, or cut may be present on the two strands. Okay, so to rectify and to repair all these type of problems, the problems that comes because of the chemical changes of the bases, or because of this chemical analogs, or because of the chemical agents like acridin, ethidium, proflavin, so such type of things. Okay, or to repair this type of breaks, to repair the this type of breaks, or to repair the thymine dimers. Okay, the cells have a very comprehensive repair system to rectify all these type of problems. And the cells have different types of you repair tools to repair this kind of DNA damages. So it's not like a single tool that can be used for to fix all type of the issues. So there are specific tools that can repair a specific type of issue in the DNA sequence. For example, when chemical changes that results in formation of abnormal bases like hypoxanthine and uracil. So as you know that these hypoxanthine and uracil are abnormal bases, so easily the repair system can identify them. And with the help of the simple base excision repair system, so initially they remove the bases. For example, when we take the structure of the DNA, so there you know that the base is linked to the sugar. Okay, and the sugars of the two adjacent bases are linked by the phosphodiester bond. Okay, so initially this base action repair only removes the base which is abnormal, or which is mistakenly incorporated. Okay, so for that we have a base excision repair system. So it removes the base initially, then also it removes the sugar and phosphodiester bond also, that means backbone also. So initially it removes only the base, so we call it as a base action repair. There is one such repair system. So there is another one is a mismatch repair system. So mismatch repair system is different from the base action repair system. How the mismatch repair system is different from the base action repair system? In base action repair system, there you find only the abnormal bases. So the abnormal bases are removed. But in mismatch repair system, for example, as I said, when guanine, okay, in amino form or keto form, when it is in a when you take the adenine and the guanine. So when adenine is in a immuno form, we know that in general it pairs with the thymine, but it pairs with the cytosine. The guanine also, when it is in a it's a keto form, it pairs with the cytosine, but when it's in a enol form, so it pairs with the so instead of the cytosine, so this guanine pairs with the thymine. So this type of mismatches, so we call these are the mismatches. So okay, we call these are the mismatches. So to remove such mismatch, suppose this is in the this is template DNA. In the template DNA, suppose it is in the immuno form and it is in a enol form, so it pairs, so the daughter DNA may add cytosine and thymine instead of the thymine and the cytosine. So to remove such mismatches, there is a mismatch repair system is present. So we must understand that mismatch repair system is different from the base excision repair. And base excision repair, the bases, for example, as I said, no, they are in abnormal conditions, so such bases are removed. But in mismatch repair system, the bases are not in the abnormal condition, so there is a mismatch between the daughter DNA strand template DNA strand sequence. Okay, not only the this tautomerization process, even as I said, no, for example, the guanine. So when the oxidation of the seventh carbon atom of the guanine, also that results in formation of bond with the thymine instead of the cytosine. And also the alkylation of this same guanine at its fourth carbon, also on its fourth carbon, or sorry, on a sixth carbon, so on the at sixth carbon also results in formation of bond with the thymine. Okay, so this type of mismatches are rectified by the mismatch repair system. So base excision repair system, mismatch repair system. And we have another one is a nucleotide excision repair system. So this nucleotide excision repair system, when DNA has a thymine dimer, suppose, so the DNA has a thymine dimer, so because of this thymine dimer, so this DNA structurally and chemically undergoes changes. So because of this chemical difference or change in the chemical difference in chemical structural difference in the bases, and the topological structural difference in the DNA, so that results in activation of nucleotide excision repair. In the nucleotide excision repair, so this nucleotide excision repair, it it's not like mismatch repair, like where adenine is present, opposite is a cytosine instead of the thymine. It's not such a problem. It's a problem with respect to not the mismatch. So it's not because of the chemical change of immunoform or some other form. So because of this thymine dimers formation, so the DNA, the topology of the DNA changes. Okay, so in such a cases, so this nucleotide excision repair system removes the part of the DNA where such type of damage is present on the DNA. So that is a nucleotide excision repair system. So base excision repair system is there, mismatch repair system is there, then nucleotide excision repair system is there. So apart from these, we have also type of repair system that is called homologous recombination repair system, non-homologous end joining repair system. So when DNA has a breaks, when DNA has a breaks on a single strand or on a two strands, so the DNA the damaged DNA is replaced by the homologous recombination repair system, or a non-homologous end joining system, non-homologous end joining system. So there is another repair system that is called a translational repair system. So what is this translational repair system? So this translational repair system is a completely different one. So when during the replication, when the polymerases cannot when the polymerases cannot replicate a part of DNA which is damaged, so this translational repair system. Okay, so translational repair system. So this translational repair system which replicates the DNA where the DNA damage is more. So when the DNA damage is more, this translation repair system replaces the regular replication machineries, replication machinery, and synthesizes DNA. And because this translational repair system doesn't have it cannot suppose DNA is highly damaged here, or suppose some chemical changes and everything are there. So such time in such condition, when the polymerases cannot proceed further, so this translational repair system, it replaces this polymerase main polymerase enzyme in the DNA replication, and it synthesizes the daughter DNA strands from the template. So because it doesn't have any proofreading ability and everything, so the reason where the DNA is processed by this translation repair system, so the reason has is a more prone for the errors. Okay, that's why it's called error prone reason. So error prone reason. So apart from all these, so we have another one that is called photolayers system. So this photolayers system is called a direct repair system. So in all the cases, whether it is a transnational, so except in the transnational repair system, when it is whether it is a non-homologous enjoining or homologous recombination repair, or a nucleotide excision repair, or mismatch repair, or base section repair. So in all cases, we remove the DNA, and we replace this DNA with the new nucleotides, or the new part of the DNA. But in the direct repair system, there we don't remove the existing nucleotides or the wrong nucleotides. Rather than we convert that existing nucleotides into the normal bases. For example, thymine dimers are there. So we don't remove these thymine dimers from the DNA, and we freshly add new thymine nucleotides to the growing DNA strand. So instead of that, we remove that covalent bonds between the thymine dimer. So when the covalent bonds are removed, so the thymine dimers become the monomers. So this is called the direct repair system. So when we say direct repair system, there the nucleotides are removed or not removed. When we say it's the indirect repair system, there the nucleotides are removed. So basically we have this type of the repair system. So here we can see the different proteins which involve in this type of repair systems. For example, mismatch repair system, their mutase is protein which scans the DNA sequence. So wherever the mutations or mismatch mutations are there, so this mutase, it searches all the DNA, mutase searches all the DNA, and mutal protein which helps the mutase. So that means it stabilizes the mutase protein, and mutH, this mutH protein, it cuts the DNA. So like endonucleases, it cuts the DNA. So after once the mutH cuts the DNA, so next that polymerase 1 enzyme, for example, so which has the 5 prime 3 prime exonuclease activity, and also the 3 prime to 5 prime polymerase, sorry, it has both 5 prime 3 prime exonuclease activity. Using 5 prime 3 prime exonuclease activity, it removes this DNA, and with that 5 prime 3 prime polymerase activity, so it extends the DNA. Finally the DNA ligase also comes. So pyrimidine dimers, so photolayase is the enzyme which removes this. So it's the direct repair system. Base excision repair, so damaged bases, so initially where the base and the sugar are linked by the base and sugars are linked by the glycosidic bond. So initially the glycosylase that removes this particular base by cutting this glycosidic bond. Later the phosphodiester bond is removed by the specific type of endonucleases. So when the phosphodiester bond is removed, the total nucleotide is removed from the DNA. Then a new nucleotide is incorporated with the help of the polymerase 1 enzyme. So that's why the polymerase 1 participates in the base excision repair also. Okay, next, nucleotide excision repair. So pyrimidine dimers, when the pyrimidine dimers, that means thymine dimers, are present, and also when bulky adducts are present on the base, in such case this nucleotide excision repair comes. So in this nucleotide excision repair also, there are proteins like UER, B, C, all these are there. So this UER A and B, so they are helpful in finding this kind of dimers and bulky adducts on the bases on the DNA strand. Once on a DNA strand these regions are identified, then URC, it cuts the DNA. Okay, Then the other proteins, so they, for example, URD, so it separates the DNA, like helicase, it separates the DNA two strands, okay, so that single strands will be formed. After that, so polymerases and all other proteins, they come and repairs that part of DNA. So in case of double strand break repair, and that is very important. So when double strand break is there, so when single strand break is there, that is very easily rectified by the this repair system. So when only single strand break is present, so for example, using the other DNA as a template, so the remaining part can be filled by the polymerase enzyme, using polymerase and ligase enzymes, and with the help of some exonucleases, with the help of the exonuclease activity and polymerase activity. But when two strands have the cuts, and when two strands have cuts, there is a problem. So there we need a non-homologous end joining. So without maybe in this process, some part of DNA may be lost by the chromosomes. So just the ends, the end part of the DNAs, they come together, and they join with each other, and that is there. So non-homologous end joining. So but homologous repair system, for example, when two strands have the cuts, suppose this type of cuts, and it's a homologous end repair system. In a homologous, there it utilizes the other chromatid as non-sister chromatid. So it uses other non-sister chromatid as a template, and so when it uses a non-sister chromatid as template, so that one of the broken DNA strand enters into the non-chromatid, non-sister chromatid. That means inversion takes place in this process. So because of this inversion, so the other other part of the strand on the non-sister chromatid, so that forms this kind of loop. And this further this strand, it uses the non-sister chromatid as template to grow, and finally this loop, this kind of loop, so this kind of loop is used to synthesize the DNA of the other DNA strand. So this is a non-homologous and homologous end recombination. In non-homologous end joining, so there the non-sister chromatids are not used for repairing. So they simply they come together, and they just join with each other. But it's when it's a homologous repair system, so single cut is there, so it uses the opposite strand as a template. Suppose two strands have a cut, so in such case, so it uses the non-sister chromatid. So that's why when we say non-sister chromatids, it is very important. Only non-sister chromatids are seen only during the S phase and G2 phase. So in the G1 phase, only you find chromatids, only non-sister chromatids you don't find. So you find chromosome like this. In the when it enters into the S phase, so it produces the non-sister chromatids. For example, this is the one chromosome. So you find so for two chromosomes you find two non-sister chromatids. So this becomes sister chromatids, and this becomes non-sister chromatids. Okay, so suppose breaks are present on these two chromatids, so present on the two chromatids. So in such case, so they uses these two non-sister chromatids, these non-sister chromatids, as a template to repair the DNA. So this is a homologous recombination repair, and the same non-homologous end joining repair systems, they work. In non-homologous end joining, they don't need any template DNA. But see, non-homologous end joining may be possible during the G1 phase, because there are no sister non-sister chromatids. You don't find it any non-sister chromatid or sister chromatids. So in the G1 phase, you find it non-homologous end joining. So once the cell reaches into the S phase and G2 phase, where if DNA has a cuts on the both strands, there you will find it a homologous recombination repair system. So this is about the DNA repair mechanisms. So probably in the next video, there we will discuss in detail each type of the repair system, like this nucleated excision repair, base section repair, or mismatch repair, and other. Thank you.